Good morning, everyone, and welcome to our UMKC Innovation Center Whiteboard to Boardroom webinar series. My name is Jim Baxendale, and I work with the Whiteboard to Boardroom program. Some of you may be new to our webinar series, and these technologies are presented by our institutional partners across Kansas and Missouri. These are technologies that are available for licensing to established companies or as new startups. On occasion, we will actually feature newly formed startup companies as well, those that are seeking CEOs, capital resources, mentors, advisors, and those types of things. Our presentations will run somewhere around 15 minutes or so in length. And we'll have some time at the end of the presentation for questions. Of course, you can use the chat feature along the way if you like. These sessions are recorded, so all of you that have attended will receive a link in a few days to that recording. And we do encourage you to share the information with colleagues, anywhere in the region or the U.S. That you think might have an interest in the technology. Today we have from University of Kansas, Dr. Wang. He's assistant professor in the School of Pharmacy and Medicinal Chemistry at the University of Kansas. The Wang Laboratory is focused on novel pharmacological modalities of modulating RNA, splicing by small molecules, also exploring new approaches to regulate innate immune response in mammalian cells. Also with us today is Dr. Shwini Betha. He directs the KU Center for Technology Commercialization at the University of Kansas. So Dr. Wang, if you'd like to go ahead, that'd be great. Thank you.
Thank you for the introduction, Dr. Baxendale, and it's an honor to introduce our recent work about early drug development of antiviral compounds against SARS-CoV-2. And next slide, please. So the first question we ask is where to target in the viral life cycle. And I'm sorry, this is a busy slide, but I will guide you through on the target selection in this slide. So, coronavirus uses the spike protein, which is a viral protein on the surface of the virion, and hijacking ACE2. This is a host protein for the entry. So this is step one. And FDA already approved 3 monoclonal antibodies targeting this process. But alternatively, when the virion enters the cell, then the nucleic acid matters released into the cytosol. So as we all know that coronavirus, this is an RNA, positive strand RNA virus. It actually has a large, about 30K BP positive strand RNA, and this is a single strand. So in a single strand RNA, all the genes is encoded in that RNA. So that RNA is 5' capped and 3' polyadenylated. So to a cell, this is a messenger RNA. The first event of the cell to treat this RNA is to translate this RNA. So our drug modality is targeting this step. So either using RNA targeting small molecules or alternatively antisense oligonucleotide. Those drug modalities are used to inhibit the translation of the RNA at the very first step. So alternatively, if the translation goes on, then the very first gene, which is a polypeptide, will be translated, and that polypeptide, either PP1A or PP1B, encode several non-structured proteins, will be translated. But before it's become functional, it has to be cleaved from the polypeptide. cleaved by the two viral proteases. And the protease inhibitors, such as Paxlovid and other compounds, will inhibit this and then kill this life cycle. So alternatively, if the life cycle goes on, then some of the non-structured proteins will reassemble to some very essential complex for the viral life cycle. So for example, the replication translation complex, or RTC. So this is RNA-dependent RNA polymerase and responsible for the RNA replication and RNA transcription to replicate the nucleic acid matters. So some FDA already approved drugs such as remdesivir or molnupiravir, so those drugs target this step. Alternatively, if the life cycle goes on, then the polymerase will further replicate the RNA and generate the messenger RNA of other structural messenger RNA. And then those structural messenger RNA, structured protein genes will be translated and then assemble the viral variants and then complete this life cycle. So as you can see, we have different points in this life cycle that we can target. What we're targeting is something new. So the RNA targeting is still an emerging pharmacological modality. So the advantage of this is that this is a brand new mechanism that no other existing drugs target at. So a hallmark of RNA virus is that it uses the RNA structures to regulate biological functions. So those structures are essential. If we are targeting those essential structures, then potentially we can avoid drug-resistant issue later on. And also, as you can see, comparing to the three existing drug targets, we're targeting something new. So it's very likely that combining the therapies will achieve synergistic effect between the RNA targeting small molecules and other drugs. So because this is a new technology, there are also some limitations. So I will state the limitation up front here. So the first one is the gene selectivity. This is because the RNA targeting and other protein targeting modalities will have a completely different spectrum of selectivity. So the key issue here is to find a molecule that's very specific for our target. And the second limitation is organ distribution, because this is a brand new modality. The distribution of the drug is still under study. So we've recently published a paper that's in the bottom right corner, summarizing all the features for the RNA targeting drugs. So please go to the next slide. So we have developed in the lab a workflow to identify the RNA targeting drugs for antiviral research. So from various infected cells, which is a collaboration with Dr. Chiu and KUMC, that we can use a method called chemical probing. And in about 2 weeks, we can elucidate the topology of the RNA. So this is much quicker than the protein structure elucidation. So the structure we're targeting is located at the 5' UTR or untranslated region of SARS-CoV-2. So this four-way helix, this is where the AUG start codon harbors. So the second step is to find the preliminary hit, preliminary ligand that binds to this unique four-way junction. So we have an in-house collection of RNA targeting compounds with existing scaffold, and within a week, we identified a hit that binds to the cavity in the center, the four-way junction. So as all the other small molecule drug development, the hard part is the lead optimization. And in our lab, we're currently doing that. And the lead optimization, we are using state-of-art RNA degrading chimera approach. So basically, we coupled an effector arm, which can lead to RNA degradation to the ligand. So this chimera is an analogy of PROTAC in the protein world, but instead we use this chimera to degrade RNA, but not the protein. Please go to the next slide. So what is our compound? We previously worked on a class of molecules called Coumarin derivatives. And recently we published a paper showing that this type of Coumarin derivatives are a versatile RNA binder. It can recognize different types of RNA structures. But this doesn't mean that these compounds are promiscuous in the structure recognition, because there are 5 rings in this type of molecules. And actually, we found that ring A and DE controls the binding affinity and specificity for the RNA receptor recognition. Please go to the next slide. So we started with a primary hit from the library. It's called I7C2, showing on the left-hand side of the slide. And again, it has 5 rings. So we synthesized more than 40 analogs of this molecule. And in this work, we focused on the DE ring. So we changed different R1 and R2 substituents. And luckily, we have a compound, an analog C30, which improved the binding affinity KD value about 6-fold from the parent structure S7C2. So all the structures contains a coumarin, which is a BC ring. So coumarin is fluorescent. So we can use the fluorescence of this molecule and use an assay called fluorescence polarization assay to accurately evaluate the binding affinity. And on the right-hand side, this is a titration of all the three of the analogs in the system. And C30 is much better, where it shows that C30 is much better than parent compound S7C2. Next slide, please. So, and then the target 4-way junction is called SL5 in literature. So SL5 is still a RNA that contains more than 140 nucleotide. So where exactly the compound binds to? This is the next question we're going to answer. So we actually split up this RNA structures into different substructures, into different pieces, and transcribe them individually in the test tube. So what we found is that the 4-way junction, when we the four-way helix, when we individually transcribe the helix, the 4 helix, the binding affinity are all low. But when we include the center junction into the SL5C, which is a very short helix, and then the KD retains as a submicromolar. So this is actually very similar to the KD of the compound C30 to the whole SL5, which is about 0.21 micromolar, 210 nanomolar. So then we concluded that the compound actually binds to the center cavity of this complicated four-way junction. Go to the next slide, please. So the next question is that how can we make use of this tight ligand to make it functional? So this is inspiring from the PROTAC modality that can translate the binding affinity into an inhibitory effect. So we need to find a good way to degrade the target using the tight ligand. So the only RNA degrading chimera in the literature is called Ribotec, or the ribonuclease targeting chimera. So we're targeting this ligand with an RNA-L recruiter. So RNase-L, this is a ribonuclease that exists in all the cells. So even in SARS-CoV-2 infected cells, RNase-L expression will not be affected. So by combining these two moiety together and form a new chimera molecule, the RNase-L will be recruited to the target and be activated by dimerization. So go to the next slide, please. So we synthesized a prototype molecule combining the C30, which is Coumarin derivative. We found a tolerable conjugation site and use a PAC linker and the published RNase-L recruiter. So based on this Ribotec, we tested the various RNA level in the cells. And indeed, this chimera can reduce the RNA, the viral RNA level in the cells. Go to the next slide, please. So in summary, we have found a new antiviral modality, and this is an analogy of PROTAC in RNA. So this is a brand new modality, and I envision that in the next decades, this can bring about the RNA targeting modality can bring about a paradigm shift in medicinal chemistry. So this is completely different mechanism to all the existing drugs targeting SARS-CoV-2, because all the other drugs targets protein and we're targeting the RNA. Potentially, the cocktail therapies can have synergistic effect. And moreover, this modality is adaptable to combat other RNA viruses or other strains of SARS-CoV-2. So we're seeking for research funds or licenses for commercialization. Next slide, please. So this is the contact information for this patent. Dr. Patha and Dr. Baxentel. And thank you. I'm happy to answer any more questions from you.
Yes, Dr. Wang, and maybe Ashwini as well, where are you in the patent prosecution for the technology?
Jim, this is Ashwini. We filed a provisional patent application recently before Dr. Wang was about to publish a paper, and it's still at the provisional stage, and we are actively marketing the invention, and we are open for either collaboration or licensing the invention to commercialize the technology.
Okay. Dr. Wang, what's the what are the next steps or further testing that you want to do either on your own or in collaboration with potential licensee? What are the next steps?
So the next step is to test the efficacy of this compound in live cells, and we have a collaboration with QMC BSL3 labs to test the real life virus in cells, and once we achieve that, then I think that's the milestone that we're considering the next step, either for federal funding or a startup company for this. Yeah,
and I wasn't sure. I was following your development of analogs, but do you have a lead compound at this time, or are you still working through that?
Yeah, we're still working on the lead compound. So right now the compound efficacy is not ideal because in virology, usually we're seeking for a compound that can inhibit the viral titer more than more than 2 logs. So in other words, 99%, this is the minimum requirement, or haven't achieved that, but we're actively doing that by using optimized effector domain for the RNA degradation. So, but we will have more progress in the next few months.
Okay. Ms. Schwinny, in terms, I heard you mention startup. Is that a possibility? And if so, what would that look like, or what would you need for that?
Sorry, I was on mute. We are open for any path to any commercialization path, either through a startup or an existing company. For startup, as I mentioned, if any entrepreneur is interested in discussing with us from the perspective of spinning out a company, we would like to discuss it. But as of now, we don't have one potential startup, but we still have that in mind with our startup ecosystem folks discussing about it.
Yeah. Okay. Okay. Are there any other questions from the attendees at this point?
Jim, I just had a quick question for the team. Have you already gone after the research funding? I assume you haven't gone after SBIR funding because you're not set up as a small business yet, but or you were talking about the next step, clinical testing that you're going to do. Is that through research funding? How are you attacking that?
Dr. Wong, have you been trying for any funding? And then I will take the other question about SBIR.
Yeah. So there are 2 NIH application pending for this project, and the result will. I will know the result in early next year. I think one application will be February. The other one, the council meeting is in March.
Yeah. Regarding SBIR funding, we have not applied for any funding yet, but once we are at a point where we have a company registered, and then probably for the April 1st or 5th deadline, hopefully. Yeah.
Wonderful. That's great. Thank you. It was very interesting.
Dr. Wong, I'm curious what an ideal licensing partner looks like. Are there any attributes of that entity that would be most interesting infrastructurally or intellectually to you as a partner?
So, Dr. Wong, the question is, would you be interested in taking part on the scientific advisory side if a licensee is available to commercialize your technology? And what do you expect for the company infrastructure, and what is the minimum infrastructure that's needed to start the company and get the research done at the company? Probably you can talk from the scientific point of view. Sometimes the companies sponsor the research back to your lab, and maybe you can use your own resources in your lab through a sponsored research agreement. SBIRs help in that type of research. So would you be willing to be part of that?
Yeah, of course. So right now, the current strategy in my lab is still seeking the federal funding for the research, but I'm happy to serve as a scientific advisory board for the company, and yeah, that's a good option too.
And what would be the minimum funding needed to go to the next step of data?
Yeah, so usually at this step, it's just like all the other small molecule optimization. When we have the lead compounds, we'll need an R1 type of NIH funding to for further development. And I think for a startup company, it will be the same. So a regular R1 type of support is needed because we need to make sure the compounds is selective and only targets the viral genes, but not the host genes. So that requires thousands of analogs to be synthesized. So that's the minimum requirement of the support for the next step.
Yeah, you can always rely on a contract research organization for synthesis of compounds to speed up things. I don't know if that answers your question, Mr. Williams, but would you like to rephrase your question?
My question was answered. I don't know if that was Chris Raycamp. I don't know if Anthony had a question as well.
Oh, I'm sorry. Yeah.
Okay, any other questions? Okay, well, if you have other questions that come up later, don't hesitate to reach out to Sweeney Betha or myself. We'll be glad to get you in touch with them and the inventor to pursue this further. So thank you all for attending and have a great day. Thank you.
Thank you for the invitation.
Thank you, Jim. Thank you, Dr.
Thanks
Wong.
so much.